The radiation is one of the biggest risk factors, and this has been well described by NASA and many other astronauts and researchers. And so there, we don't have to just measure the radiation or just look at DNA being damaged. We can actually actively repair it. This happens naturally in all of our cells. There's little enzymes, little protein, and really many machines that go around and scan DNA for nicks and breaks and repair it. We could improve them. We could add more of them, or you can even activate them before you go into space. So we have one set of cells in my lab where you activate them before we irradiate them to actually prepare them for the dose of radiation. And now that is what's called epigenetic CRISPR therapies, where you can actually, instead of adding or taking away a gene or modifying a cell, you just change kind of how it's packaged. Like I was just describing, that the DNA, the genes are still there, we're just changing how they get used. And so you can actually preemptively activate the DNA repair genes, and we've done this for cells. We haven't done this yet for astronauts, but we've done it for cells. And a similar idea to this is being used to treat sickle cell disease and beta thalassemia. As you can reactivate a gene that was dormant as a way as a therapy. For copy, it's good to just have one big statement, but you want to do both. Or a third option, I would want to, do electromagnetic shielding. I would want to do a fourth option of, maybe some other kind of physical defenses.
So outside of the human body.
Yeah. So we're taking the same passion to keep astronauts safe that's outside them and just putting it in their cells is what I propose. Now, it's a bit radical today because, we're just starting this in clinical trials to treat, diseases on Earth. So it's not ready, I think, to do in astronauts. But in the book, I propose by about the year 2040, that's when we'd reach this next phase where I think we'll have known enough about the clinical response. We'll have the technology ironed out. That's about when it's time, I think, to try it. If you really want to activate genes, like I was just describing, or repair a specific disease gene, you can actually CRISPR it out, modify it. This has been already published and well documented. But as I was alluding to, more and more we'll see people that you just want to temporarily change your genes' functions and change their activity. So the best example of this is for beta thalassemia. We all have hemoglobin in our blood that carries oxygen around. And when you're an adult, it's a different version. It's a different gene. You have one gene when you're a fetus called fetal hemoglobin. When you're an adult, you have a different gene. But they both are making a protein that carries oxygen. After you're born, the fetal hemoglobin gene gets just turned off. It goes away, and you replace it with adult hemoglobin. But if your gene for hemoglobin is bad as an adult, one of the therapies is, well, let's turn back on the gene that you had when you were a fetus. And it's actually already led to cures for sickle cell and beta thalassemia in this past year. So it's this extraordinary idea of like, well, you already have some of the genetic solutions in your body. Why don't we just reactivate them and see if you can live? And indeed you can. So I think we'll see more of that. That's for severe disease, but eventually you could see it for more, I think, work-related purposes. Like if you're working in a dangerous mine or in a high radiation environment, you could basically start to prime it for work safety. Basically, we need to genetically protect you. Now, it would have to be shown that genetic option is safe, reliable, that it's better, that at least as good or if not better than other shielding methods. But I think we'll start to see that more in the next 10, 20 years. And eventually, as I describe in the book, you could get to recreational genetics. You could say, well, I want to turn some genes on just for this weekend because I'm going to a high altitude, so I'd like to prepare for that. And so instead of having to take weeks and weeks for acclimation, you could just do some quick epigenetic therapies and have a good time in the mountains and then come back and turn them back off. If it's just in space, it'll be hard because you'll need basically some gravity at some point. I think you'd need orbital platforms that give you at least some partial gravity, if not 1G. If you're on Mars, it's actually, even though the gravity is 38% of Earth's, just having that gravity would be enough. And if you could get under the surface into some of the lava tubes where you have some protection above you from the radiation, I think that would be, you probably could survive quite well there. So I think it's just in space part that's hard. You'd need some gravity, you need some additional protection from the radiation. Plants have been grown in flight and you can get some nutrients, but right now it is very reliant on all the up mass being sent up, all the freeze-dried fruit that then gets rehydrated, which doesn't taste awful, but is not self-reliant. Right? So I think those would have to be small bioreactors. It'd have to be a lot of work on fermentation, a lot of work on essentially prototrophic organisms, the organisms that can make all of the 20 amino acids that you would need to eat. I describe a little bit in the book, what if we did a prototrophic human, where you could have, like right now we need to get some of our amino acids because we can't make them all, which I think is kind of sad. So what if we could make all of our own amino acids or all of our own vitamins? Also, I think that's one case where another adaptation would be to activate the vitamin C gene. Like right now you'd have to have limes or some other source of vitamin C in space. but we actually carry the gene inside of our genome to make vitamin C. Look at dogs and cats, for example. They have these kind of wet noses. You don't see them going out and getting margaritas, although dogs can drink beer and get drunk. They don't need vitamin C. They have no risk of scurvy because they can make the vitamin C all by themselves. So can other wet-nosed primates called Strepsirrhines. But we are dry-nosed primates, and we lost this ability. Sometimes 10 or 20 million years ago, we no longer make our own vitamin C, but the gene for it is called gulo. It's still in our DNA. It's what's called a pseudogene. It's just broken down. It's like having in our genome, we have these functional genes, like a nice BMW, a nice car that works well, but we also have this junkyard of old cars, old genes, old functions in our DNA that we could bring back. And so vitamin C is one of them that would be very easy to do. So then you could activate the gene, repair it so we can make our own vitamin C. Now we'd have to do it again carefully, because what if we lost vitamin C, the production of vitamin C as a species? What if it was a good reason that we lost it? Maybe it was helping in some other way that we can't see now. But you'd start slowly, do it in cells, then do it, potentially in animal models and other primates, and then try it in humans. But that's something else I'd like to see. So we wouldn't have to make as much food in orbit. You could actually start to make as much of your own food in your own cells. But I don't want to be a robot. Humans love, as I do, texture. I realized that made me sound like I wasn't human, but humans love food and flavors and textures and smells. All that is actually attenuated in flight. So you'd want to not forget our humanity and sort of this love of all the benefits and wonder of food and cooking and smell. And they've announced they want to have boots on the red planet by 2033, which is 2 to 4 years earlier than when NASA's supposed to do it. So we'll see if they get there first. But I think it's a space race 2.0. But it's not just the U.S. and Russia this time. It's China, it's India, it's the UAE, it's Europe, ESA. JAXA has the Japanese Space Agency, and there's the U.S. So now it went from just a 2-person race to a whole field. The whole field of runners, if you will, on the track trying to get to Mars first. And I think it's gonna be like anything. If you start to have settlements and construction projects and places to visit on Mars, I think that the true mark of a place being actually settled is when you start to be able to pick. You're like, well, I want to go to this destination, not this one, because they have better Martian cocktails here, but this one's not as good. So that this idea of innovating and competing will continue to drive, I think, humans as they always have. The number of missions planned, there'll be several missions per year through multiple space agencies and companies that are really making low Earth orbit very routine. And by go up, I think it might also, for example, the Inspiration 4 mission just went up for 3 days in flight. And it was enough time to get up there, do some experiments, enjoy the view, and then you came back. The Axiom missions are a bit more complicated. There's docking, you're up in the space station, it's a shared atmosphere, so you have to follow all the ISS protocols. What's interesting about the Dragon capsule and the Inspiration 4 and some of these what are called free flyer missions, you can just launch into space. You basically have your own little mini space station for a few days. It's not that big, right? But I think that's what we'd probably see him do first, because we're going to see a lot more tests of those in the next 2-3 years, but they're already been demonstrated to be safe. And then you're not trying to go for 10 to 20 days or months or years at a time. It's just up in space for a few days, but you're in proper space. It's an orbital flight. It's not just a suborbital flight. You could do the podcast from there.